All right, I think we're good to go. All right. Again, hello, ladies and gentlemen. Thank you for joining us today. My name is Sebastian Küchenmeister. I'm a marketing manager here at Certara, and I have the pleasure today to open the webinar and to introduce our expert, Kevin Trimm. Hey, Kevin. I will hand over in a second, but please let me first provide some housekeeping items for today's event. This is a webinar of about 30 minutes. Kevin will provide input and will also show us the tool and demo. Towards the end of the webinar, you have the opportunity to ask your questions. If you experience any technical difficulties, please message me using the chat. As a reminder, today's webinar is being recorded. We will email you after the event and send you the link to the recording in the next few days. During the presentation, all participants are in listen-only mode, but you have the opportunity to submit text questions using the chat pane in the control panel. So, as I said, we will answer your questions right after the main presentation. And that was it for the moment from my side. And without further ado, Kevin, the screen is all yours.
Thank you so much, Sebastian. So welcome all. It's my pleasure to speak to you today about the newest version of PK Submit 2.0. Today we'll go through a demonstration helping us to see how we can create complete pharmacokinetic regulatory submission ready CDISC domains in just a few minutes. So although it's possible to do it in a few minutes, there is quite a lot of functionality in PK Submit 2.0. So I will take much more than a few minutes to go through this so that I can explain all of that functionality. And it may in fact go more than the 30 minutes that we intend because of all that functionality. But if for any reason we don't have enough time for all the questions at the end, we will of course be happy to respond to all questions by email. So for the session, like I said, we'll just go through a demo of a typical PK workflow from import of the source data right through to the end of the generation of CDISC domains. And we'll go through an overview of all the new features included in 2.0, and there are many. So quick introduction to me, I'm the VP of Product Management at Certara. I've been working in this field since about 2004. Where I've been, studying and working in pharmacokinetics, using really Win-non-Lin and eventually Phoenix that entire time. And I've been at Certara since early 2018, where I've been a product leader, helping to introduce some new products within the pharmacokinetics area, and that includes PK Submit. Okay, just a kind of overview of what PK Submit is all about. So it is really, one of the main purposes is really to save development time. Certara has a mission of accelerating medicines to market, and PK Submit is one of the ways that we can help you to do that, because it really does shorten the time associated with creating these electronic data submission files. It really allows the PK analyst to generate those files as a part of performing the usual NCA workflow. It reduces the risk and errors that can be introduced through a more manual process, which helps protect you against audit, so that if you are audited, it's you have a clear and traceable workflow for how these domains were created. It optimizes R&D productivity. So like I said, you can allow the PK analyst to be the person who's actually creating these domains without necessarily learning a lot of information about CDISC and data standardization. They can really just focus on the core competencies of performing the NCA and have those domains created as a byproduct. And one other thing worth noting here today is that we have integrated the Phoenix Win-non-Lin and PK Submit with Pinnacle 21 community version, which is just another saved step in the validation of those domains, so that you can have the gold standard Pinnacle 21 validation report once again from the community version that allows you to see very quickly any problems that might be associated with the domains that you've created. This is very helpful when you're passing those domains over to your data management group for the preparation of the submission. Okay, so I mentioned that there are a lot of new features with PK Submit 2.0, which is certainly the case. So let's kind of go over some of them now before we jump into the demo so we know what we can expect to see. So we do have the ability to adjust date, sample, and day times to the pharmacokinetic profiles directly within a UI. We have enhanced merge functionality and summarization with data checking, sort of validation of data as we're loading it post-merge. We have the ability to specify which samples are trough samples so that we can flag them for reporting purposes or for exclusions. We have the ability to implement complex BQL substitution rules, more than what is present in Phoenix today. We have the ability to augment data and add missing key analysis variables that might be there. We include a user interface for baseline correction, dose normalization, and also sample duplication. So these are, complex data preparation activities that we often have to do. We've got a graphical user interface to facilitate those things. We allow for exclusions, including graphical exclusions, conversions of units, which is more sophisticated than what we've done before. We've enhanced the slope selector to make that process much more easy and seamless. We've have an enhanced ratio functionality now within PK Submit that allows you to make more bespoke ratios in a more straightforward manner. Added compliance for all current SEND and STTM implementation guides. So we have 3 implementation guides per standard now within PK Submit. We have editable expressions for the domain generation. So we make the way in which the variables in SEND or STTM, the way in which they're being generated, more transparent and customizable if necessary. And finally, we do have that integration with Pinnacle 21 to produce the validation report, but it's also this community version of P21 that's going to be producing the data reviewer's guide and the define XML file. So that's why I'm concerned that we may need more than 30 minutes. All right, so let's go ahead and get into the demo. So we'll start with Phoenix, and we will go ahead and create a new project in Phoenix. And once we have a project, we will import some data. And so I'm going to choose an EX and a PC domain. So of course, PK Submit will create a PC domain, but often we do find that a PC domain is in fact the starting point in which analysts are working with. So I'll use that as an example. I should mention that it does not require that you start with a PC or an EX domain. You can really start with any kind of data set that you want and then proceed in using PK Submit. So now, because I have a license for PK Submit, I do have this menu item in my version of Phoenix, and I will begin at the top of the menu in the data preparation wizard. So here I have the opportunity to load my data, and it's looking in my data folder and it's finding those 2 data sources, which I can choose to load. I don't have to choose I could have any number of data sources in here and I don't have to load all of them, but that's where it's going to be looking for the data. And I have the ability to specify what type of data set each of these is. So the EX domain is obviously a dosing type of data, whereas the PC domain is a samples kind of data. We also recognize demographics data as well as ADCL, the subject level atom data, that sometimes is interesting in order to have all the variables that we need for the NCA. So these are the sort of out of the box categories that you can assign that help simplify the next step. So let's move forward with this. And now we enter the part of the application where we are going to be performing a merge of the samples and the dosing data. You know, there's things that we need from the dosing data to perform the NCA, not least of which the dosing levels themselves. So we need to perform a merge, and we need to specify which are the variables that are going to be used as the sort keys to perform the merge. So one thing I should note, and maybe we'll switch over here to the samples data. Within PK Submit, we do have a data model that is, you know, native to the application, where there are a number of variables that we are either expecting or reserve a position for, so that we can proceed with the analysis. And so because I've loaded a PC domain and an EX domain, PK Submit does recognize the variables present in those domains, because those are known data set types. If you have a data set from Watson or some other place, you can manually specify here which value should correspond to the internal data model. But in the case of loading CDISC domains, PK Submit already knows that model and is prepared to perform the mapping automatically. And so the subject, the samples data is the data in which we'll merge the dosing data into. And for the dosing, we just simply have to specify which are our merge keys. So subject is an obvious merge key, but we're also going to use analyte and day as additional merge keys. And you can see we have the auto mapping here as well. Always at the bottom of the screen, I'm just going to make this a little bit bigger. We do have the preview of the data, both for the dosing as well as the samples. And we have this neat little feature over here on the side that allows for day and time adjustments. Often when we receive PK or PC data, the labels for day don't correspond with how we'd like them to for the analysis. For example, a 24-hour sample might be labeled day 2 because technically it is the next day, but we don't want to treat that as day 2. We want to treat that as day 1. And so even though the day variable may codify it like that, we have the opportunity here to make that adjustment and to edit the analysis day. In our case, the alignment is okay. We don't have this issue, but it's worth mentioning this functionality and to show that we can do the same for the time as well. So regardless of what the nominal time might be labeled as, if we need to adjust the nominal time that we're going to use for the analysis, we do have the opportunity to do that here in a very user-friendly and straightforward manner. Okay, so now that we have our data loaded and mapped, we're able to move forward and perform the merge. So this will just take a moment while it's merging. Just bear with us for the merge to complete. Okay, so now we've completed. All right, so the first thing we see is a merge summary, so that we can really see the source of, you know, what data set was merged into the samples data set. In our case, it was doses. We can see the original name, the number of rows that were within that data set, and the number of rows that those correspond to in the samples data set, which is obviously much more rich because we have multiple time points per occasion and only one dose, so it's 416 to 64. But now we've created equivalent dosing records for all those 416 from those 64, and everything was found, and the status is green and successful. If there are discrepancies, you can have warnings and color code of yellow. And if, for example, one of your merge keys was empty, you can actually have a red and to tell you that the merge in fact failed. But in our case, everything merged together very well. We have a preview of that merge worksheet, and we actually have a detailed log of all the merging activities. For those people that would like to see this, especially it's more relevant if you do have problems with your merge. This log will help you perform that troubleshooting exercise. So let's move forward. And now we have some preliminary validation. This is a sort of health check of the data, where we have a number of rules set up that we've categorized as either high, medium, or low, where it's going to interrogate the data that you've merged together, really just looking to see that we have all the necessary variables that we're expecting in order to eventually create the PC and PP domains. So we do see down below that one of our rules has been flagged, and that is because our time unit is not valued. So within the data that we loaded, there was no time unit, and so that's going to repeat for every record because it is required for every record. And so we're going to have to ameliorate that whenever we move forward here. But now we know that something critical is missing because of this validation, which is very helpful. All right, so I'm going to click submit now. And as a result of that, I do have now a merged result. So this is the result post-merge that is a combination of my EX and PC domains that I loaded at the beginning. Okay, so now we've completed the first part, the first menu item, this data preparation wizard, and we can move forward into the analysis wizard. And the first thing we see in the analysis wizard is kind of a data wizard. If you're familiar with Phoenix, this is a little bit like the data wizard object, where we can apply expressions to the data to augment our results. So I'm going to do that now because I have to rectify the fact that my time unit, which I can see down here in our handy data preview, is not valued whatsoever. And so let's refer to the time unit column, and let's put a very simple expression here where I'm simply going to specify that I want to value that column with this value here. And I can execute that. And now I have it. You can do much more complex things, because of time constraints. I'm not going to get too sophisticated with all the things that can be done here. It is the same sort of Microsoft syntax that you're used to with the Phoenix Data Wizard. There is the syntax reference here. If you click, it will open this help site for PK Submit, where all of the syntax is described here so that you can build whichever kind of expression you like. Okay, so for me, simply populating the time unit with hour is sufficient. So I will move forward and come to the screen where I will be identifying my my profiles for the NCA. So these are the sort keys that you need in order to perform the NCA to group the data appropriately. So we have some default sort keys, which are always appropriate, even if they're singular in nature. It doesn't harm to have them. So, subject, analyte, matrix, and analysis day. We do use the terms of our internal data model rather than whatever the variables were called. In this case, CDISC naming. We're not using those. We're using the internal ones because there could be any variety of data sources. I can add additional sort keys if I like. For example, dose, perhaps that's needed to make sense of that. This is just warning me that if I've set any trough values, they'll be reset by changing the variables. That's perfectly fine. So now I've got dose and I've added a variable here. You can see these are my NCA sort keys across the top, and now I have dose as well. And that's been included as a grouping variable for my NCA. So this is a preview that's very nice to have because I can click on these groupings and I can see individual profile, but with the time points included for each of these profiles. It's really a list of all the profiles in the study, and if you can see here, it tells me the number of samples, and so I can see which ones are really troughs and which ones are actual pharmacokinetic profiles, and I can have a look to see what those data are like. We always like to have these data inspectors within PK Submit. So I mentioned that we clearly have some trough samples here, which I might like to tag as a trough sample. And so the other thing we do within PK Submit is we allow for this filtering of the data. So I can filter on the number of samples and choose the value of 1. So I can reduce my list to just those so-called trough profiles. And if I click Control A, I can select all of these profiles and do a right click and say, set these as to trough samples, which auto ticks the box for me so that I can identify samples as trough samples. Now, why would I want to identify these as trough samples? Probably I'd like to exclude them from my NCA. I also might want to use that trough data set as for reporting purposes. So I can make, for example, a plot that's showing all my trough samples over the course of the study separately from the actual profile plots. So we allow you to make that indication here. Okay, you can also come in here and set the sampling design. This is a serial study, so I'll leave it at that. And I can move forward now to the next screen. I'll go ahead and maximize this again as well. So the point of this screen is to apply some substitution rules to non-numeric codes that are found in your data. Obviously, the most common non-numeric codes that we encounter are codes that are indicating less than a quantifiable concentration from the bioanalytical method, less than LOQ or BQL or BLQ. However, the lab has made that code, and we allow you to substitute those values as you wish. There's an object in Phoenix that allows for BQL substitution as well. This one is a little bit more sophisticated, and that we allow for both unconditional and conditional substitution rules. And for the condition, we allow for 2 different reference points. So in Phoenix, I believe it's really just the Tmax that allows you to reference, but we can also here do the first quantifiable, which seems to be the more commonly used rule. So as part of our data model in PK Submit, we will be producing 3 new concentration columns. Obviously, you've had a concentration column that you loaded and you mapped to the internal variable, but we're going to derive 3 new columns, and we allow you to specify different rules per column. So just to kind of understand the intention of these columns, the PK concentration column would be the one that you would normally use for the NCA itself. but we allow you to have a separate column that you might want to use for plotting and another column that you might want to use for your tables. And so these, these variables are created so that when you complete the process of PK submit, they're available to you for the usual Phoenix reporting. Okay, and so for unconditional substitutions, maybe for the plots, we want all the non-trough samples converted to 0. And then perhaps for our PK concentration, we want those values before the first quantifiable treated as 0, but then after the first quantifiable, we're going to leave it as actually missing. So I'm just going to leave that blank and it'll convert it to missing. And perhaps for the display, we actually don't want any rules applied because we actually want to see those values in the table. So we can execute that, and now all of those rules have been applied to this data set within these concentration columns here. We left the original concentration untouched, but you can see the sorts of substitutions that are happening over here. Okay, so let's move forward. And this is our dosing screen. So one thing we do have to indicate here is the dose type. This will drive the type of model that's used. And since I can see that my data is organized here by analyte, route, and matrix, I can choose what dose type I would like to use for that combination. We will show here all unique combinations of those 3 variables that will allow you to select per combination. So this is just to make this more straightforward. We also allow you to set the molecular weight for your analyte if you know what it is. The reason why this can be helpful is when you're making ratios downstream, you may want to correct, for example, a parent to metabolite ratio by the molecular weight. That won't be relevant for us because we only have one analyte, but very useful nevertheless. Okay, so what other information can we enter in dosing? A lot of this information will have come in with the dosing file that you merged early. However, if you don't have dosing information, you can still proceed. And when you get to this screen, you can include that dosing information if for any reason you didn't have it. And of course, you're always in a position to augment it. So for example, tau, I can add at this time. Tau will drive whether it's a steady state or a single dose type of NCA. And so perhaps here I can use my filters to say that I'm going to add tau to these 2 occasions. And so now I can enter this value of 24. And I can drag down to populate that and then clear the filter. I'll just clear the filter. And you can see now that's been applied. Something else that's also handy here that becomes relevant downstream is to actually go ahead and indicate which is my control group. So here, perhaps I can use my dose variable and say, show me only the 0 values, and then I can say that this is a control group equals Y. And I can do that here as well. And infusion duration, similar sort of concept if you have an infusion study, which I do not in this case. There's another tab here at the top that allows you to calculate all your concentrations, the dose normalization of all concentrations. So you simply indicate which is the dose column of interest, which is the concentration column that you want to base this normalization on, and I can click this button and then get this variable, which now will appear here down at the end, which I may want to make some plots, for example, with these dose normalized concentrations. So I have the ability to create that here. Okay, we'll move forward. And now we're at a calculation screen where I can calculate a few things, including time deviations, as well as baseline corrections. So let's do a quick example of a time deviation. I'd like to specify one starting at time 0. and going up to perhaps 4 hours. And I'll change that hours, and I'm going to make it less than or equal to. And I'm going to allow for a 15-minute deviation or a 10% difference. And if I fall outside of that range, I'm going to flag that as a time point which has deviated. I can add a second rule where I can say greater than 4 hours and up to and including 24 hours for the second part of my rule. And now I will increase the limit from 30 minutes to 30 minutes and perhaps 15%. And I can execute these rules. And that will allow me to have deviations applied. So if I look at my filters, these filters are always very useful. I can see I didn't get any hits with those deviations. They're all values of 0. There's no differences between my actual and nominal times. And so I have a very clean sampling schedule here. But if I did have deviations, they would be flagged and I would be able to leverage those flags downstream. So let's take a look at the baseline corrections. So for each analyte, I can set up different baseline corrections. And of course, baseline corrections are very useful if we are dealing with drugs which happen to also be endogenous molecules, for example, hormones, and we are needing to subtract sort of baseline values from the values that are associated with the exposure. And so we have the ability to apply several methods to do that here, where I can use a fixed value, I can do a pre-dose correction, which is like a within subject correction, or I can leverage my control group to generate a mean by time point where I'll do the correction, or perhaps just an overall mean of the entire group over time to derive the mean to perform that subtraction. This is why it was useful that I could, in the dosing screen, specify what my control group was so that this utility could reference that group to derive those values. I don't have an endogenous molecule here, so I'm not going to do any baseline corrections, but this is the way that you can make those specifications and perform that correction. Okay, so now I have a sample duplication screen. So often nowadays in both preclinical and clinical trials, for economic reasons and for limiting the invasiveness on the subjects participating in the study, often pre-dose samples are not collected during the repeat dose phase of the study or trial. So in order to complete the profile, we would like ways to estimate those concentrations at time 0. And so we have a number of different methods here that allow you to do that, where you take an observed value that was collected under another label and insert it at time 0 according to some logic. And so a very popular one to use would be to use this rule, which is called use last sample of previous adjacent profile. And what we mean by that is, for example, with a large molecule study, you might have a sample taken at 168 hours after the first dose, so-called day 8. And then you may have another profile that's being collected on day 8, but now you're not going to take a pre-dose sample because that 168 sample was already taken. And that is the time 0 sample of dose 2 on day 8. So one sample can only have one identity in a LIMS system, but we allow you here to duplicate that sample to use it under 2 different identities. So this one is always a good rule to apply if it's valid for the study in sampling design. But we have other rules as well, like to use the C-tau or the C-last or the C-min to estimate the value at time 0. This use C-min is the default rule of the NCA engine in with non-lin from the beginning. So this is something that would actually be applied anyway during your multi-dose part of the study. But you have the, we allow you to choose it explicitly here so that it's intentional and clear on what's being done. So I can execute this as well. And you can see I actually got some hits, right? So I've actually inserted a few samples or records, I should say. I'm inserting some records where I've duplicated the sample from the previous adjacent profile to estimate or to actually represent time 0 on those occasions. So this is very handy. This is a complicated thing for you to do using the Phoenix workflow, and it's been greatly simplified through this user interface. Okay, so now I'm coming to an exclusion screen where I can exclude some of my records from analysis. When I'm excluding, I have to indicate what I'm excluding from. Typically, it's the NCA. We do allow you to exclude from the PC as well. Perhaps you've loaded some data which really don't belong in the PC at all. We've seen examples where you might have some titer information from anti-drug antibodies or something like that mixed in with your concentration records. And so you have a mechanism to really drop those completely so that they don't appear in the PC where they're not meant to be. Any exclusion that you specify, you can allow that flag to carry forward into your PC domain if you want that. That is a practice of some companies for Cent. Not everybody's doing that. There's some debate about whether or not that's appropriate. We're not entering the debate. We're simply allowing you to specify whether you want to do that or not. So exclusions are based on variables, right? So I can, for example, look for my, well, I should go back to my deviations. You can see deviation rule applied. This is related to my time deviations. So I can look for that. That's not going to be relevant to me because I didn't have any rules applied. There were no hits based on my criteria, but perhaps I could go to my trough samples and say trough equals Y and just put the reason of trough sample and exclude. And I should have some hits in here, which I'm not seeing. Exclude, let's do control group. Control group equals Y and placebo, exclude. And here I can see some hits. Probably my trough is not using the Y as the flag. I'd have to double check that to see why that one didn't hit, but this is a good example I can show here for the control group. All the ones that I added the Y myself to indicate the control group, I can actually exclude those from the NCA. Okay, any exclusion criteria that you set up, you can save and reload later so that you can reuse criteria that you specified before. You can also add comments to the data rather than simply exclusions here. So I'm just going to select a random record here, add a comment, and say just example comment for purposes of the demo. And you can see now what I've created down here at the bottom is the beginnings of a comments domain because we can actually create a comments domain that would presumably be merged into the comments domain for the entire study once handed off to the statistical programmers for in creating the submission data sets. So we can begin. If we're adding comments to PC or PP, we can put those in a comments domain format for the sake of the submission. Something worth noting here as well is that we're also able to do visually based exclusions. So for example, if I click down on my my grouping according to my NCA sort keys, I can see all the subjects that fall within that grouping, and I can click on the subject, and now I can perform an exclusion right here graphically. You know, this could be helpful to view, to help me think about exclusions. Here I see a value that's maybe a little bit high that I find suspicious, and so perhaps I'd like to exclude that from the NCA because I believe this to be an aberrant value for whatever reason. So I'm going to say, let's exclude that from the NCA. And I'll just say that that's an aberrant value in my opinion. And now I can come here and exclude it. And so that will be referenced here as well, back on the main grid. So that exclusion is applied here. And if I scroll through, I should be able to see that as well. Okay, so that's exclusions and comments. Let's move forward to the next screen, which at this point is really just providing the rest of the information that's needed for the configuration of the NCA. We do allow you to set the interpolation method per combination of the sampling design and the matrix. So it's the usual options that come with the NCA, as well as the model type as being either plasma or urine. And we allow you to select the time and concentration variables that we default to what we presume you would want to use, but we do allow you to change those as well for the NCA. We do, of course, allow the specification of partial areas as well. This is simply a grid that we're organizing using the sort keys to allow you to select the times that you want for partial areas on a profile by profile basis. And of course, you can use these filters to simplify your selections. And so you can just do the drag down by grouping things together in a logical way. We do have a nifty feature here that allows you to specify the renal clearance partial areas. That presumes that you have urine data and all the interval data that's needed to do that. So we'll just go ahead and based on when the intervals are, select the appropriate plasma partial areas so that you can get renal clearance downstream. So this is just sort of an easy button to find the partial areas in your plasma that are appropriate for your urine intervals to get those renal clearances. I don't have any urine data, so I'm not going to bother with that. We have here the ability to do rather complex unit conversions, based on, for example, a matrix, and it's showing for this analyte, which again, I only have one in my data set, it's showing me the native units, but I can change these units to, for example, microgram per liter, and I can add that as a unit conversion, and I can create a whole list depending on how many matrices and analytes I have, and I can do the same with time. And of course, I have a time variable now because I included it, so it's showing me that it's currently hour, but if I wanted to change that to minute, I could convert it at this point. Okay. I can specify some carry-alongs. So these are variables that I want to carry through even though I'm not using them as part of my NCA sort keys. I can bring them along for the ride. These are things that we're going to automatically carry, but you can add more. So if you want more things to carry through to be available to you in the data sets for use in reporting, then you can also specify those here as well. So now I'm going to click submit, and I have now an NCA object that is completely configured for me, and I can run that object to get the beginnings of my PP domain. Okay, so now I've done that. We're all familiar with the native functionality of Phoenix. And now I can come here and I can do my slope selector. For example, if I don't like the selections that were made by the NCA, which I can see here in green, I can choose different slopes. So obviously the NCA object already has a slope selector. We've enhanced that slope selector. These options are all things that you're used to seeing there, so I won't explain what these are. They are present in Phoenix. We're just, including them in this UI. But the part that is an enhancement, I would say, is the ability to look at both the exclusions in the grid and the plot at the same time, or the slope indicators, which may include values to exclude. And you can, but it's the ability of how they're organized here, where you can more easily find the slope that you're looking for rather than the tabs that exist in the NCA engine. This is just a much easier way to navigate through the slopes according to the sort keys and see them and return to them if you want to change your mind on any selection. So this is an enhanced slope selector that we have included with PK Submit. So let's go back, and now we're going to finalize our PK data, and it's going to auto select the parameters worksheet that it's finding in my NCA object. And I'm just trying to be aware of the time, so I'm going to move a little bit more quickly through this section. So this is simply the opportunity to select which PK parameters we'd like to choose for our analysis. We have some that are sort of auto-selected, but you can choose whichever parameter you like. And you can also save and load lists, right? If you have a list that you like to use, you can select it once, save it as a list, and then reload it at a later time. We have this new ratio specification where we have allow you to set up multiple kinds of ratios. For example, I might want to use the analysis day as a ratio variable, where now I can select both a reference and a test value. So perhaps I'd like my reference to be, my reference to be, let me undo that, day 37, which now removes 37 from this list because it's the reference value. And I can use, for example, 38 as the test, and I can do multi-select in the case of the test values. The sort keys associated with the ratios will by default be whatever my NCA sort keys are, minus if I've selected one of them up here as a ratio variable, and obviously then it would be excluded from this list. But otherwise, it is the same as the NCA sort keys. But if I'd like to change the ratio sort keys, I can do that as well. I can have individual values for my ratios. I could also have mean values, and I could also, instead of presenting them as a ratio, I could present them as a percent. So there's really a lot of functionality here. It's indicating to me that I haven't chosen a parameter that I'd like my ratio to be based on. So let's maybe choose AUC last, try again. And now I can see that this is a ratio that will be calculated. It's an accumulation ratio, and we do have a list of prefixes here that you can choose from to help you keep track, and you can also just make your own. I'll use one of these pre-indicated prefixes that will be applied to my AUC last ratio based on individual values on those 2 days compared to day 37. We can also allow for creating concentration ratios. It's not only for parameters, concentrations can be here as well. Same sort of idea, obviously, that you're not choosing the parameters though. In this case, you're simply choosing which concentration column you'd like to use. So I'll click OK. And my ratios will be specified, and I can see I have the ratio here now as a parameter that I've selected. I'll click next. Once again, we have an exclusion screen. Very similar concept. I will perhaps show that I can set up criteria here based to exclude a parameter based on the value of another parameter. So for example, percent extrapolated greater than 20. The reason of terminal phase not reached. And I can actually put a replacement value for that into the data as well. So instead of seeing the value and it being flagged for exclusion, I can actually choose to replace it. And so now I've applied that exclusion set. I'm not sure if those criteria were relevant for my data. Looks like I have nice terminal slopes across the board. So I'll go ahead and click submit. And that will finalize my PK parameters. And so now I have a PK parameter set that's ready to be converted to PP, along with a concentration data set that's ready to be converted to PC. And so I did load a PC data set here, but PK submit will create its own PC data set based on all the activities that I've done up to this point. So we return to now we're into the level of creating the CDISC domains. So let's go ahead and do that. Where I have the opportunity to choose which standard I'm trying to use. So I'm going to stick with send because I do believe these are animal data. And I'm going to choose send 3.1.1. We do have, as I mentioned before, these are the implementation guides that we allow you to comply with, which is right up to date. For the SAS transport files that are created, I actually have the ability to choose which version of SAS transport file is used. V8 is the usual version, but there is a newer version of V9, and I guess there's still some people using V5 from many years ago. So this is the standard that we'll use. And this is the PC domain, and these are the expressions associated with that domain that that PK submit will use to create the domain. There are C# expressions here explained and made apparent and how these variables are being created. The variables that don't have expressions created with them are free text, and there are other which are simply references, which we also allow you to change. If you need these other ones changed, this is a support ticket. We're not sort of assuming that the users know how to create C# expressions. It's not the same as the Microsoft expressions from the data wizard. So if you, for some reason, don't like the way that we've configured this, it would be a support ticket to help you change those values. Okay, so that's our PC domain. We have the same thing again for the PP. We have the same sort of idea, and you can see all the parameters and what this PP domain will look like based on those expressions. You have these previews. So let's go ahead and submit those. And now we have a PC domain, a PP domain, a rel rec as well that will tie together the PC and the PP. We create a sub PC as well as that comments domain because I did in fact make one comment. So we do have that now as well. So the domains are here. They're not yet in the final format, but they're there for preview. So let's create the submission files. That's the next step. And for the define, there's some information that we can specify. So in the interest of time, I'm not going to do this, but this is just information that gets included in the define file. It's not present anywhere else in the data to this point. The define file would not be complete without this information, so we do prompt the user to allow them to enter that information. So we're going to go ahead and create the define file. It's going to be in an XLS or XML format. Just takes a moment to generate. So if we look into our, give it a second there to complete. if we look inside of our documents folder, we can see now I do have XPT files for all of my domains that were derived from here, and these are ready to be submitted. I have a defined file as well, and I have an Excel version of a defined file. This is, sometimes asked for by the your biostats group or whoever it is that's making the rest of your electronic submission. So that's now available. And I can come back to say, let's submit this, those XPT files to Pinnacle 21 to get our validation report as well. And so this is the same sort of interface that you get from the community version. I should update here to say I'm being compliant with 311, and I can have my control terminology selection there as well. So now that job is being submitted to Pinnacle 21, and we'll get finally a validation report from there. Okay, so if I look inside my documents, I have a NSDRG. This is non-clinical study data reviewers guide. And inside my Excel, this is my Pinnacle 21 validation report, which I can view through the external viewer. So I can see I've got a couple of items here that are saying there's some problem in the global metadata that would result in a rejection if I were to submit it to the FDA. But I can see here that these are just the fact that our demographics domain and the TS domain are not present. That's fine. I didn't intend for them to be present. I'm not submitting at this point the entire submission to Pinnacle 21. I've only created the PC and PP. So you can see there's some issues here with other domains that are not present as well. But otherwise, not looking so bad. A few issues down here with the PC or PP, which are, we won't have time to drill into these, unfortunately, to take a look at those issues, but this is a standard Pinnacle 21 validation report that will be very familiar to anybody who's used to making these domains. So I'll go ahead and close that. And the only thing left to do within this is really to export those SAS transport files that are ready at this point that can be used for submission. So that brings us to the conclusion of our demonstration of PK Submit. There's an awful lot of functionality in there. So we did take much longer than I think we sort of thought we could, but I did want to make sure that I could go through all of it because there is an awful lot of new functionality compared to the previous version. And certainly if you're not familiar with PK Submit, there's an awful lot there compared to Phoenix itself. So Sebastian, I'm not sure, do we have maybe time for one question before we conclude?
Yes, we have actually one question, and that is about how the new version of PK Submit is compatible to Phoenix. What versions does the new PK Submit support?
Okay, so PK Submit is going to be compatible with all the supported version of Phoenix. So that goes back to 8.1 at this point. So it is compatible with all the supported versions. That's typical with all of our plugins. If it's before that, we would encourage you to update Phoenix anyway, so that you are on a supported version. But all of our plugins, we do make them compatible with whichever versions are currently under support.
Okay, great. Thanks, Kevin, for the answer. That was the only question we had so far. And with respect to your time, I would suggest that we answer all your other additional questions by email. If you have more questions, please reach out to your account manager or to sales@certara.com, and we will answer these questions as soon as possible. And with that, I think we can wrap it up for today. Again, thanks, Kevin, for the presentation. Thank you for joining us. As I said before, we will send the recording of that webinar to you in the next